So welcome back, my dear children. I hope that you have watched my previous video and have noted down notes as well in your notebook. So today let us start oogenesis. So let us see what is oogenesis. So it is a process of formation of mature female gamete. This process initiated during embryonic stage. See, oogenesis starts from, or you can say starts at the age of embryonic stage. Okay, so in next image I will show you how this develop. Okay, how oogenesis processed. Then during this stage mother cells are formed. Okay, that is known as your oogonia are formed. So once the oogonia are formed at this stage, what happened? It get arrested. So at which stage it will get arrested or stop, we will discuss later. So after that, what happened? No more oogonia are formed, okay, and added after birth. So once the parturition takes place, there is no formation. So no more formation of oogonia takes place, okay. So in last class, I taught you spermatogenesis, right? So oogenesis is unlike spermatogenesis, okay? They are not similar. So spermatogenesis starts at puberty, but oogenesis starts at embryonic stage when it is at fetus, okay? So in next picture, I'll show you how the oogenesis starts at embryonic stage. Now let me explain you the process of oogenesis, how it starts at embryonic stage. So here you can see it is a fetal ovary, okay? It is the ovary of a fetus, alright? So here at the beginning, the fetus ovary has a medulla, cortex and oogonium. Okay, so small dot that you can see are cells. Okay, oogonium is also known as germ cell. So what happens is, first this oogonium, it undergoes mitosis differentiation. See, as I told you, that differentiation means what? When one type or one form of cell, it is just modified into another type, okay, and division is or reduction where it get divides, all right. So I hope that the differentiation and division, the concept is clear to you. So then what happens? This oogonium, they undergo differentiation process and they form primary oocytes. All right. So this germ cell, okay, oogonium, it will become what? Primary oocytes, which is diploid in nature. Now, then also this process does not stop there. Again, this primary oocytes, they will undergo mitosis. Okay, and again they will form what? Primary oocytes. So here what happened? At the stage, primary oocyte doesn't change, as you can see, okay, but what changes takes place is the cells which is there in a cortex, it will start, okay, this primary oocytes, it will start to collect them, okay, it will get surrounded by them, and these cells are called as your granular cells, and the structure they form is primary follicle. Am I clear? See, this is how. So this is how the development of follicle and oogenesis are related. Okay, you will learn this in step by step. See, first what happened? There was only oogonium, the germ cell. It undergoes differentiation, so it forms what? Primary oocyte. So after that, mitosis takes place in primary oocyte. So primary oocyte remain unchanged. Okay, but what changes takes place is that it is surrounded by some cells, okay, and those cells are called granular cells, and as a result they form a structure called primary follicle. Am I clear? Okay, now here what happened? The process of oogenesis stopped, okay, because after that delivery of baby takes place, parturition takes place. See, where does it get arrested? At primary oocyte, when the germ cell become primary oocyte, and it arrested it at prophase 1. Okay, meiosis, the meiosis was about to start, but the process get arrested. Okay, and that is, we know that in meiosis there are different stages, anaphase, metaphase, right? So prophase, so it get arrested at prophase 1, which is also known as diplotin stage. I hope that you are able to follow the sequence, that germ cell, it undergo differentiation from primary oocytes, then primary oocyte undergo mitosis. After mitosis, primary oocyte will remain as primary oocyte, but primary follicle is formed, and it has entered meiosis 1 stage, but it does not complete meiosis 1. So it get arrested there at prophase 1 and is also known as diplotin stage. So what happened after that? Parturition takes place. Okay, now parturition to puberty. This period, after the birth of a baby and a puberty, okay, no oogenesis takes place, means no more formation of oogenesis takes place. So here the meiosis 1 get arrested, means it got stopped there only, right? But during this stage, what happened? Follicle, which starts forming here, right? After mitosis in a fetal embryo, the follicle which has started forming starts degenerating. Okay, and when it reaches to puberty stage, okay, while reaching that stage, the follicle started degenerating, and only 60,000 to 80,000 of follicles remain per ovary. Okay, in one ovary, means in each ovary only 60 to 80,000 left. I hope that you can follow me till here, right? Okay, now let us see what happened next. After here, the process has stopped, okay, and there will be no more activity between parturition and puberty stage. Okay, so let us see after the formation of primary follicle what happens. So at puberty stage, what happened, as I taught you earlier, that hormones are secreted, right? So GnRH, I hope you remember, this gonadotropin hormones are secreted, which take parts or which initiate the formation of this ovum, right? Okay, now we learned that primary follicles has developed, fine. Now what next? Now this primary follicle will later transform into secondary follicle. Now how does it develop? What happened? So after that, what happened? So this primary follicle, okay, so here the primary oocytes, okay, it does not change, it remain same, but granulosa become multi-layered and surrounded by theca. So here you can see the diagram, right? So earlier there was only one layer of granular cell, correct? But now this granular cell has become multi-layered, and one more layer has formed. Okay, the outer layer has formed, or a layer which is surround the granulosa, that is called your theca. All right. Okay, now this starts when a female, okay, when it reaches to its puberty stage. See, when she was at embryonic stage, till then only the this primary follicle has formed, okay, and it has arrested there, okay, at prophase 1. Now the period between the parturition and puberty, okay, before she has reached puberty, there was no activity. Now once she has reached the puberty stage, hormones will secrete, okay, and it will initiate the oogenesis process. And then what happened, that primary oocyte will transform into secondary oocytes. They will start dividing. Okay, the divisions takes place, the meiosis division, right, it start. All right. Okay, now because of that division, this granular cell, it will start forming a double layer, multi-layered, okay, and a theca will form. So the formation of theca and multi-layered granulosa uh, is an identification of secondary formation of secondary follicle, right? Okay, now next what happened, once the secondary follicle has formed, then comes your tertiary follicles. Now, what happened here? As I told you, the division starts, right? So, in tertiary follicle, due to the division, okay, meiosis, what happened? The primary oocytes, okay, it divides into two cells. There was only one cell, right? Now this primary oocyte will divide into two cells. Now out of that two cells, you can see one cell is at this inside, right, the ovary. So one cell that is which is inside the ovary is called your secondary oocyte. All right. See, primary oocytes divides into two cells. One forms secondary oocyte and other you can see at the external layer, that a small cell that is called your polar body. So primary oocytes, when it enters into the tertiary follicles, here what happened, the meiosis process completes. Okay, the meiosis 1, it completes division, complete division of cells, and it formed two cells. One is secondary oocyte and then polar body. Other than this, there are few changes that takes place in ovary, that is a cavity like structure, okay, starts forming, and that cavity is filled with fluid, and that cavity is known as your antrum. I hope that you can follow this. Okay, in secondary follicle, there you can see only one layer of theca, isn't it? But when it enters or when it transforms into tertiary follicles, this theca will divide into, or it will form more than one layer. So the inner one, which is more close, okay, the closer theca is known as your theca interna, and the external one, the outer, which is little far away, is known as your theca externa. I hope that this is clear. So let me give you a quick revision on this. See, what we learned in previous is that when the primary follicles are formed at embryonic stage, there the process stopped. Okay, no more formation of oogonium takes place. So when it reaches its puberty stage, hormones are secreted. Okay, gonadotropin, GnRH hormones are secreted, which initiate the oogenesis process. Then what happened? The primary follicle, it's transformed into secondary follicle. And what are the changes that takes place? Is the multi-layer granulosa and a theca is formed. Now, what happens is next, this secondary follicle transformed or developed into the tertiary follicle. Now tertiary follicle, what are the changes taken place is that meiosis 1, which was stopped at the early stage, has again started at puberty stage, and meiosis 1 is stopped, has completed, okay, is completed in tertiary stage, and because of that the division of cell takes place. All right, and it forms two. The primary oocytes will divide into secondary oocyte and the polar body, and because of this, unequal division takes place. So because of unequal division, the secondary oocyte is larger inside and polar body is smaller in size. Okay, then cavity is formed, which is called antrum. Theca becomes two layer, theca interna and theca externa, and the secondary oocytes, okay, is surrounded by the zone pellucida. So the secondary oocytes is surrounded by the zone pellucida. All right, I hope that this is clear to you. Now let us see what happened once the tertiary follicle is formed. So after the formation of tertiary follicles, so once the tertiary follicle is formed, then again this tertiary follicle will transform into graphene follicle. So graphene follicle or mature follicle. Then at the last what happened, this matured follicle, they will further ruptures, means they will break down, okay, their external layer will break down and they will release the secondary oocyte from ovary. So secondary oocyte is what? Ovum, okay, is the egg. So it is released. So I hope that the process of oogenesis is clear to you. So if you have any doubts regarding this, just let me know, okay. So the process that I had explained you is summarized here, okay, and it is categorized into different phases, that is multiplication phase, growth phase and maturation phase. So here you can see the primordial germ cell, okay, here you can see they just multiplied, right, they grows into so many oogonia, they develop or they form so many oogonia, and one of them is grown, okay, so this is called your primary oocyte. Then later this primary oocyte will undergo the first meiotic division, well they will get divided half and one of the body is bigger than the other body, okay. All right, and then the ovum is formed and there you can see additional polar bodies are formed. Okay students, let me tell you one thing. In spermatogenesis, 4 spermatozoa were formed, right? But here only one ovum is formed. So this is again one difference between spermatogenesis and oogenesis. So I hope that you are able to follow this. So when you are watching my video, then do keep your NCERT book along with you and compare your textbook writing with my explanation, okay. You just compare both together, then this will help you to understand better. So let us see how spermatogenesis and oogenesis are different from each other. Spermatogenesis is a process of formation of sperm. On the other hand, oogenesis is a process of formation of egg. Spermatogenesis, it occurs in male gonad, testis. Oogenesis occurs in ovary. In spermatogenesis 4 spermatids are formed, and in oogenesis only one ootid is formed. Spermatogenesis is initiated by testosterone and oogenesis is initiated by estrogen. In spermatogenesis no polar body is formed, but in oogenesis 3 polar bodies are formed, and the growth period in spermatogenesis is short as in comparison with oogenesis, okay. In oogenesis it is little time taken, it's longer in oogenesis. All right, so this is all for today's class. So in my next class I shall teach you the next topic, that is menstrual cycle, okay. So this is all for today. Go through this video and take down the notes. All right, so I'll see you in my next class. Till then, take care.